Proteoform and isoform both refer to different versions of a protein, but there's some subtle differences between them. The key differences are that an isoform can refer to different versions of a protein that actually come from separate genes, whereas with a proteoform, here it's all coming from a single gene. But with a proteoform, you incorporate post-translational modifications, so you count different post-translationally modified versions of a protein as different proteoforms, so one that was phosphorylated versus non-phosphorylated, or phosphorylated at this site versus this site. These would all be different proteoforms, but those would still be considered the same isoform. So let's think about where all this variety can come from, starting from the gene and then going all the way to post-translational modifications. So the gene itself, you can have variations in the gene. So different people can have slightly different versions of a gene. We often talk about SNPs, or single nucleotide polymorphisms. And so this is just a single letter change at one site in the DNA, which may lead to a change in the protein letter that gets made or things. And so that would be an example of a SNP that's providing some genetic variation at the level of the gene, and that would influence the proteoforms. However, we would still typically just refer to these as the same isoform of a protein if they were just slightly different versions in different people. But if you have multiple copies of a gene in a single person, then they were slightly different. Those would be considered isoforms, but not proteoforms. Typically, there's some exceptions, but that's the basic rule. We often refer to this type of isoform as a homolog. And so if they're coming from the same gene and then that gets duplicated and they evolve separately, we would call those paralogs. So those would be isoforms. Those would not be considered, however, proteoforms. With the proteoforms, however, and the isoforms, now you go from the gene and you need to actually make this into messenger RNA. RNA polymerase can start at different sites, so you can have alternative promoter usage. And if it starts here, you're going to get a longer pre-mRNA than if it starts here. And you can also have differences in where it kind of stops. So where do you add that poly A tail? You can have different polyadenylation sites that are then going to make the end of it shorter or the end of it longer, and all of those are going to contribute to variety at the level of the pre-messenger RNA. And so those sorts of changes, differences in the promoter usage and the adenylation site usage, those would both be considered differences that can contribute to the isoforms or the proteoforms. So those can provide variety at the level of the pre-mRNA. And then that pre-mRNA gets spliced to form the mature messenger RNA. You can have alternative splicing that leads to the inclusion or the exclusion of specific exons, and that's going to provide even further variation at the level of this alternative splicing. Now, these changes that we were just talking about, this alternative start sites, the alternative splicing, alternative polyadenylation, those would be considered contributing to both the isoform and the proteoforms. So now we're at the level of the messenger RNA. We've got the ribosome going to translate and make the protein. Often we think about, oh, there's just that one start site, that one AUG. Well, sometimes the ribosomes can actually start at different sites, and these aren't always even an AUG. You can have things like leaky scanning. You can have alternative, like upstream ORFs, so open reading frames, various things like this. The ribosome can start at different places, and therefore you're going to get different length of protein products. And those would be considered isoforms and proteoforms. But now you've got your finished protein. The finished protein at this point, with the isoform, we're like, okay, we don't care about anything that happens from now. This is still the same isoform of the protein. So you put on a hat, who cares? Now, however, we have the proteoforms. The proteoforms will still say, okay, well, if you put on a hat, well, now you're a different proteoform. And so if you phosphorylate this site, you're one proteoform. If you phosphorylate this site, you're another one. If you phosphorylate both of these, you're a different one, and that sort of thing. And you can not only have phosphorylation, you can have ubiquitination, you can have acetylation, methylation. There's all these different variations of a protein that you can have after that protein is already made. And all of those would be considered different proteoforms. So there's this vast number of proteoforms, and that's actually really hard to know how many proteoforms there are. Proteoforms are inherently difficult to measure, and so there's a variety of ways that people try to measure the variation in the proteins that get made, but each of these has kind of pros and cons. You can get at the genetic variation at the level of the gene by doing DNA sequencing, but that doesn't even tell you if that protein actually gets made. To see if a protein gets made, then you're often going to look at the RNA. When you do just general RNA sequencing, you typically do these short reads where you get these little pieces, and so you don't know how things were hooked up in terms of were they alternatively spliced. So this was coming from this messenger RNA, but this was coming from this messenger RNA, and now we can't tell which came from which. And so we lose that information about splicing. And so that's one challenge. There can be overcome by kind of longer read sequencing, but then again, we're thinking about, okay, well, we've got these messenger RNA products, but are they actually being used by the ribosomes? And so then you might look at things like ribosome sequencing to see which are actually getting used by the ribosomes. But even if you get that information that the ribosome was on there, that doesn't necessarily mean that we're making a functional protein product. Even if you take the assumption that they are making a functional protein product, they're just making that kind of like one protein, and now you need to think about, okay, well, what about the post-translational modifications that get added onto that protein to make it a different proteoform? And those modifications you're not going to know about just from that RNA sequence. So now we need to actually go and look at the protein itself. The way that this is typically done is going to be through mass spectrometry. Typical mass spectrometry is what we call bottom up. You digest the protein, or basically use enzymes to chop it up into little pieces, and then measure the mass to charge ratio of each of those pieces, compare those to a database of pieces from different proteins, and therefore figure out kind of where the pieces were coming from. Now we're getting back to that problem we had, though, of when we were doing the RNA sequencing with these short reads, is that we can't tell what goes with what. So did this phosphorylation site here correspond to the same proteoform as the one that had the phosphorylation, this peptide that had the phosphorylation at this site, or are these from different proteoforms? So we lose that information. An alternative, therefore, is top-down proteomics. And so here you actually go and you measure the intact protein, and then you kind of break it down and look at the individual pieces. Now, this is going to be like more technologically challenging and stuff, especially if you have a bigger protein, and you still have the challenge that the modifications have to survive that processing in order to get them. And so sometimes with mass spectrometry, say, when you're doing the ionization points, when you're trying to like get the pieces to be charged so you can go see them, it's going to actually modify the protein, modify the modifications, and so you can't get an accurate idea about the proteoform diversity. And so you kind of have to take all these different layers of information and try to figure out what proteins are getting made and what proteoforms are being made from those proteins. And so there's a lot of complexity, but scientists are trying to actually map all this complexity and catalog it in a human proteoform database. And so it's pretty cool. And what they're trying to do too is determine some of the combinatorics at these different levels. And so differences in splicing, say, leading to the inclusion of an exon that now has this site that can get phosphorylated. And then that phosphorylation can have differences in the activity of the protein. And so you have all these factors that kind of build together these different layers of variation that ultimately are leading to this vast diversity of proteoforms. And then they're trying to catalog all these proteoforms in the human proteoform database, as well as try to incorporate information about different disease states, say, that correspond to different proteoforms. And so lots of cool stuff to keep your eye on, but hopefully this also helps you see not only how incredibly diverse the protein population inside of us is, but how complicated it is to actually see how diverse that protein population is.